I'd like to welcome you to day two. Also thank our sponsors that helped fund this event. Things like food, some of this future travel. It was very helpful to have people help with that. Here. So we greatly appreciate that. This to preview today. Also to remind you that in about 3 to 4 weeks, the slides will be made available to participants. Also, we're going to prepare a proceeding. So if anyone wants to contribute to that, contact myself or Dana, and particularly Dana, are more responsible. But anyway, today we're going to have kind of 3 sessions and then a wrap up. We're going to start morning talking about excipients. This is obviously a very critical issue to the formulations. So then we're also going to talk about patient centricity and then imagining the future. And then finally, as I said, a wrap up. So this is a preview for today. And that will wrap up. So and with each of these, we have a panel session. So the first session is with the excipient, and here is the outline for that. So we'll start off with Ndidi Tiwari, who represents the industry, and also to remind you that we have all of the detailed bios online. So if you want to see the detailed bios, and he represents the excipient companies, those perspectives. Then we'll have Braddy talk about application, and then Ndidi going to talk about some of the, as a reviewer of excipients, some of the issues that they face with the FDA, and then one second. I hope people get here. I'll work with data. All right. Anyway, they're working on that, some of the audio issues. And then finally, we're going to have a breakout session with Anjali and Smita. We're going to talk about the software that be used to help with pediatric workplace. So this will be our main focus for this morning. So without further ado,
Deep's going to give us the
like.
Yeah, just here's a laser.
This one.
What?
This.
Yeah.
Good morning.
Morning.
Yeah, thank you, Professor Hoag, for that kind introduction and kickoff of the session. I just wanted to give a little bit of a background as to how I and also the excipient industry sort of got associated with this sort of an initiative. So the story started in 2022 when Professor Daniel Schofferberger invited me to speak in AI/ML workshop focused on pediatric research at Johns Hopkins in 2022. When I attended that workshop as a representative of excipient industry, I was amazed. I was shocked. I was sad. At the same time, I was happy as well. And I think you will understand why I'm saying this, because when you see, sometimes you wonder, very little has been done in this area of pediatric research. Whereas when you look at folks like Melanie, like what she was presenting yesterday, lot is being done as well, right, by individuals. Still, there is a lot that needs to be done in future. And one critical segment, which is very critical for this initiative, is excipients, the excipient industry. What can an excipient industry do to help resolve some of the challenges? But then here it is chicken and egg problem. And you will see that why that problem exists. Why the industry is hesitant, not only the excipient industry, but also the pharma industry. And then it creates that vicious cycle that nobody wants to take initiative and nobody wants to move the needle because there are reasons for it, right? So I'll talk about excipients in pediatric formulations. Basically, 20 minutes is not enough to give you detailed information. So what I will do, try to make it a little bit interesting, try to make it thought-provoking, and so that when you leave, you start thinking about it, right? That is my goal. My goal is not to give you a lecture in the area of excipients, because that we can do separately, right? I just want to make sure that when you leave today, you go with some thoughts. And that's why I will talk a little bit about evolution, means history. I'll talk a little bit about current use and look at future perspectives. Okay. Again, a standard disclaimer, these are all my views, not the views of my employer, BASF. We are going to look at very briefly about historical lessons. They are so critical. Many times we miss history, and if we don't know history, we don't know why things are the way they are right now, and we don't know how to shape the things in future. So I think we'll look at history, which is very important. And I have taken the advice of Professor Hoag not to make it very comprehensive. We only have 20 minutes. So I'm going to keep it very brief. Again, one very important distinction, I think we need to all understand how are excipients perceived by industry, how are they perceived by FDA, and how they are perceived by general public. I think it's very important to understand that. And I think there's very nitty-gritty detail. I think we say that devil is in the detail. So once you see that devil, you will understand why I am saying this. And then, of course, pediatric age groups. I think there are many stalwarts here. And I think Smita and Anjali are going to talk about pediatric dosage forms. I think Professor Daniel Schopperberger also will talk about it. We'll look at current regulatory expectations very briefly and then closing perspectives. So I think once you look at excipients, right, FDA, children, and historical perspectives, I want to combine this story with excipients as well as drug product, not necessarily leave it to excipients, because you cannot separate drug product from an excipient, right? So when you look at the history itself, it's very interesting. The history is tied in part to the excipients, and you will see why I am saying this. When you look in history and the origin of FDA, right, how FDA was initiated. So it's very interesting that in 1886 or 1890s, most of the formulations that were marketed at that time were patent era medications. Patent era means not necessarily patented, they were almost never patented, but they were trade secrets that they would not disclose anything about the product, how they are made, what are the ingredients. They will just be claims on the product. And one such product was Wilson soothing syrup. And this soothing syrup was used for colic babies, babies that cry a lot. It was used for neonates. It was used for large group of young infants, right? And this is the picture you see from a trade card in 1886. You will be surprised that this, along with many other formulations that were sold in the market at that time, led to multiple deaths of neonates And that created a lot of hue and cry, and that public hue and cry sort of also contributed to the formation of Food and Drugs Act, and in fact, FDA in 1906. You'll be surprised at that time, because they were patent era medications, it was not mandatory to write the ingredients on the label. So what this law did in 1906, it required the labeling. You know why? Because this product was made with morphine, and it was also made with ethanol as a solvent. And ethanol is very toxic to infants or neonates, because ethanol is metabolized by alcohol dehydrogenase enzyme, and that enzyme is not very well developed in neonates. And then it can lead to toxicity, it can penetrate into blood-brain barrier, it can lead to a lot of secondary complications, and ultimately, it can also lead to death, right? So because of that, it was made mandatory that there should be no adulteration of the food or misbranding of the food, and there was a requirement for labeling. They should be labeled, that's it, that was the only requirement. You should label. And then when you see what happened in 1912, there came an amendment, because a lot of these companies were selling these products because they were just labeling. You have to only label, nothing else. They were selling these products with the claims, or sometimes like poems. Oh, mothers, give this medication to your child. It will bring strength, it will bring energy, it will bring tone, any kind of claims, just to promote their drug products. So this amendment came in 1912 that prevented the false therapeutic claims, that you cannot have false therapeutic claim, that's it. And when you go further, the sulfanilamide tragedy. I don't want to go into the details, because yesterday John from FDA already clarified how this tragedy happened. I want to go into the excipient part of it, right? So it is very interesting, 1937 sulfanilamide elixir was made and marketed in Tennessee. But if you go back to the history, why this formulation was made, it is very interesting. Sulfanilamide get discovered in 1935 or 1930s. And at that time, like the sore throat infections were very common. April, May, like, when you have that springtime, it's very common. So the company thought, oh, the sulfanilamide is available in tablet or other dosage forms. And when you have staph throat infection, particularly for children, the company thought, oh, maybe solution or suspension will be very effective. So they advised the chemist to develop a formulation. Now the chemist developed the formulation using diethylene glycol. He used diethylene glycol because it gave desired solubility, and it also gave a sweet taste, little bit sweet taste. So he found it very palatable or acceptable for children, right? And then the formulation was made and distributed, and we know the history, like how many deaths occurred. Almost most of the product was recovered. But when you look at the details in the devil part, how this causes toxicity is very interesting. When you take diethylene glycol, it gets metabolized by alcohol dehydrogenase into various acids, glycolic acid, glyoxylic acid, glycoraldehyde, and then it causes metabolic acidosis. Now this diethylene glycol is an osmotic agent, right? So when it is rapidly absorbed, what it does, it creates edema. Number one side effect is cerebral edema, right? So it can cause cerebral edema that can lead to seizures. Number two, because it is forming oxalic acid can react with calcium in the blood-brain barrier, and you can have calcium oxalate crystals that can lead to seizures, right? So that is the first CNS side effect you see. Then secondary, you go to the heart, because there is a calcium and oxalic acid, it forms calcium oxalate crystals, and you can have prolonged QT interval. So secondary effects are related to heart complications. And third and most important is, because there is oxalic acid, it can also react with calcium in the kidneys, and it can form calcium oxalate and block glomerular filtration, leading to nephrotoxicity. And most of the deaths ultimately happen because of nephrotoxicity. So from 10 hours to 72 hours, this is how the progression occurs. And this is all related to excipients and the way the excipients get metabolized by the body. It's very important to understand that this basically set the tone for evaluation of excipients in the pharmaceutical drug products in 1938. And that is when the law gets passed, which is Food and Drug Cosmetic Acts, where it was very clearly specified that drugs need to be shown safe at this point. Until then, nothing. They are saying it needs to be safe, and then safe tolerances set for unavoidable poisonous substances. This is the first reference to the safety of excipients in the whole history of pharmaceutical drug product development. Right, and again, I think there are many other details, but that are not relevant here. We know the thalidomide tragedy. I will not go into the details of that, but basically this tragedy led to making sure that drugs are not only safe, but they are effective as well. The effectiveness came only in 1962. Earlier labeling, no false therapeutic claim, safety, and then efficacy came only in 1962. And when you go and particularly look at children, now I think this is the part which is very important for us to realize. And I realized then, when I spoke first time at Johns Hopkins, is that when you look at this timeline, before 2002, only 20% of the drugs that are used actually were labeled for pediatric use. 80% of the drugs that are used were off-label. Off-label means they were never studied for children, but anyway used because there is nothing else available, and doctors were using it, even still using it based on their experience, right? And I think why that is so, because children don't fall sick. You don't need medications for children except for cough, cold, and vaccines. So why would companies develop them? They would not. And that's why any legislation that was targeted for pediatric dosage form development was stalled until 2002. It started in 1991, but there was lobbying, there was opposition, there was whatever you can call it. It never materialized until 2002 when the law came called Best Pharmaceuticals for Children. That was a carrot from the FDA to the pharmaceutical industry that if you develop products for children, we'll give you exclusivity. Then FDA realized that carrot is not useful because many people will not take carrot because there is no money there. So then FDA brought a law in 2003 that called Pediatric Drug Research Equity Act, where it made mandatory for the pharmaceutical companies to evaluate application in pediatric population if the drug is proved to be useful in pediatric population. And that was called as a stick. So FDA made it mandatory that if the drug is found to be useful in children, it must be studied. There is no exception, right? So that law came only in 2003. So if you see the whole pediatric initiative, pediatric dosage form, I think only a handful of very dedicated researchers that you see in this room yesterday and today are actually devoting their time for this. But you will still see there is a lot of gap. And you'll be surprised that the timeline that takes from human drugs to pediatric drugs, even the clinical trial, the gap could be anywhere from 10 years to 15 years. So when the drug gets launched for adults, it's only after 10 or 15 years the clinical trials may start for children, not necessarily they start. And that is only because it is complicated, right? Many things for children are very complicated because you have the concept of informed consent, right? And I think when you ask a child, you have this medication, there's a possibility that this thing will happen. He says, I don't want that. I'm fine with how I am, right? Many times. So I think you see that, regulatory wise, there are many challenges, and those challenges are also linked now to formulations, and those challenges are also linked to excipients. Because I think I remember yesterday, I think Trupti was mentioning that we need to have new excipients, to resolve the challenges of drug product, what we have. And many times we also say that you cannot address today's formulation challenges with yesterday's excipients. But reality is different, and reality is linked to, I think, something more serious, and we'll see that why. So I think excipients are very important because we need to have palatable and acceptable dosage forms for children. Children cannot take huge tablets. We need sprinkles, we need solution, we need suspensions. But if you see the excipients which are commonly used for those excipients or those solutions or suspensions may have severe toxicity. Now, if BASF or any other company wants to bring a novel excipient, then there is a challenge. And the challenge is because FDA considers excipients a little bit differently, and I will show that in my next slide. I think it's very important to understand the definition of excipients for general public by FDA and also by industry. So when you look at International Pharmaceutical Excipients Council, they define excipients are substances other than pharmacological active drug or a product. Whereas FDA says that excipients are known as inactive ingredient, means any component other than active ingredient is an excipient for FDA. But FDA, more importantly, and I think this is the most important definition here, is that FDA considers both active ingredients and excipients or inactive ingredients as drug product components. So you have drug as a drug product component and you have excipient as a drug product component. And the implications of this definition are huge. Why the implications are huge? Because FDA puts same standards they have for API for approval of excipients in a drug product. There is no regulatory pathway for approval of a new excipient. It has to be used in IND. It has to go through NDA route, new drug application, and then only an excipient can be approved for that particular route of administration. Otherwise, there is no pathway. I think Joanna mentioned yesterday that FDA initiated a novel excipient pilot program in 2022 to give at least okay for clinical trials of excipients, novel excipients. But that program was terminated in April 2024 because FDA found that that program was not in line with the priorities of the FDA, right? So then this is a challenge. Any new excipient you want to use, it's a novel excipient, and FDA, by this standard definition, because it is drug product component, they expect the cGMP procedures for API to be similar, like API and excipients to be similar. They expect the ADME safety and toxicity studies for drugs and excipients to be similar, right? Otherwise, they would not approve easily any novel excipient. And there's a history because diethylene glycol story happened. FDA never wants to repeat that kind of a story again, right? So then this creates a challenge for companies like BASF. For example, we brought in a new excipient called Soluplus in 2009. Today is 2026. We still don't have that Soluplus in any of the approved drug product in the US. We have that Soluplus approved globally, but not in the US. So the company will not be able to materialize the commercial benefits of that investment of 20 years if they invest into novel excipient research, because there is no regulatory pathway, and the pathway is limited because if I go with a novel excipient with a company, they will say that, oh, I have this new drug. The new drug is very promising. If I bring in your novel excipient, they will study, FDA will study it in detail. My application may be delayed, may be denied. Why should I take a risk? I don't want to take a risk. I will go with approved excipient. I will find alternative approaches. I will do permutation combination of all approved excipients and see if I can make it work rather than using a novel excipient, right? So this is now preventing innovation in the area of drug delivery, dosage form, and also development of suitable alternatives for children. And I think when you look at, again, another complication here is that when you look at children, US FDA and EMEA, they have different age groups defined for children. For example, this is a table from EMA. They have almost 3, 4, 5, 6. They have 6 categories, and I believe US FDA has 5 categories. So there is lack of harmonization even there as well. So if you look at preterm newborn infants to newborn infants, and these different age groups, children, for example, and these are the suitable dosage forms, you will see that obviously for infants and toddlers and newborns, solutions, suspensions, and effervescent dosage forms or sprinkle dosage forms are preferred. You have to also remember that when a child is really small, it's very difficult for them to swallow anything. You cannot give a pill, you cannot give even a mini tablet that might get stuck. So you have to have a solution or a suspension, most preferred, or sprinkle or something like that. For that, you need suitable excipients, right? And because these excipient choices will support the swallowability, the palatability, and how the children can accept the medications, right? And I think we saw some excellent examples. So there are like different dosage forms, and one of the dosage form may not suit for the other age group. Like I think children which are like bigger children or grown-up children's, they may accept orthodispersible or chewable tablet or even a capsule, but you cannot have the same dosage form for a neonate or a small children. And that is where then you have to be very cautious when you consider new excipients or even the traditional existing excipients. And you will see why in next slide. I'll just give you one example. I think ethanol we already discussed earlier. Ethanol is a very good solvent. A lot of drugs that are not soluble can be solubilized in ethanol, and then you can easily use it. But ethanol, as I explained earlier, cannot be used in neonates. Their neonates, their development of liver and liver enzymes is very in the early stage. They do not have a lot of enzymes that are required for metabolism of ethanol or even for a lot of other excipients developed fully, and then that can lead to toxicity. So even though ethanol is very suitable, it cannot be used in certain age groups. Same propylene glycol. Propylene glycol is an excellent solvent for liquid formulations as well as for suspension. But then because it is osmotic agent, it can also lead to a lot of complications in children, particularly in neonates and young infants. Again, because of the immature metabolism. Now, this is very interesting. We want to make solutions. Now, for solutions, if you want solutions, you want to make sure you use some antimicrobial agent because you want to preserve it as a preservative. Benzyl alcohol is a very good preservative, widely used, but benzyl alcohol has a metabolism issue. Benzyl alcohol gets metabolized to benzoic acid, then it conjugates with glycine, and then it gets converted into hippuric acid, and then the hippuric acid is eliminated through urine. But in children or young children, this hippuric acid metabolism doesn't happen. Benzoic acid gets accumulated in the liver, and it can cause acidosis, it can cause respiratory distress, and it can cause ultimately death as well, or CNS complications with benzyl alcohol. So remember that benzyl alcohol is a very good preservative. It's used in solutions, suspensions, but you have to be very careful if you are going to use it for very young infants. Now, polysorbates, polysorbates are very good surfactants, benzoates, preservatives, selected sweeteners, even sweeteners, because children population, you need sweeteners, right? Aspartame or synthetic sweeteners or other sweeteners, they can cause age-specific tolerance issues and metabolism problems because I think a lot of people said it, children are not young adults. Children are children. Their bodies, their development of physiology is entirely different. It's not fully developed. And many of the things are still immature and still getting developed. In fact, I think I was just reading, I'm not sure if it was about benzyl alcohol or one of the preservatives that was being used to clean the placenta when the placenta was cut, and then, when the newborns are born, you cut the placenta and you probably sterilize it either with benzyl alcohol. I think it is benzyl alcohol, and that also led to severe toxicity in infants. Right, so I think, so these are the challenges now. When you have these challenges, now you look for solutions, you have to look for solutions within the existing approved excipients. And that is the challenge, because you may not have solutions. And you have to look for novel excipient or new excipients, but then approval is the challenge because somebody has to be willing to work with you to get their drug product approved with that excipient. Otherwise, we'll be struggling with chicken and egg problem all the time, right? And companies are very hesitant to invest in novel excipient research until they have a partner because they want to protect their R&D investment, right? So again, like how is the excipient risk evaluated today? Again, I think, we are going to have a talk on ELSA tool. And I think John also mentioned about ELSA tool yesterday. So IID is not enough. What FDA looks is that, okay, I have IID, but I also want to see which age group it is used, what dosage form it is being used, and most importantly, what application. Is it a cough syrup or it is an anti-cancer medication? Because the evaluation might be different based on the need, not necessarily based on the application. Right? So that's how FDA looks at it. And I think you can correct me if I'm wrong. And assessment depends on the route. I think this is what exactly I explained. And in most cases, the youngest relevant subgroup becomes the most conservative benchmark for the FDA. They will look for application in the youngest age group because that is the age group expected to be not having the fully developed organs and metabolism pathways in children, right? So again, this pediatric excipient is not necessarily binary. It has to be justified by age, exposure, and clinical context. I think this is very important. And again, I will not go into the details. I think John also explained very nicely yesterday about STEP and PARA framework, which is used today. And I think we are also going to hear about ELSA tool. So what FDA is doing, FDA started this initiative of ELSA, which is AI-based tool. Remember that FDA has a bunch of applications over the period of several years or decades. Now, if an FDA reviewer wants to review particular excipient, imagine the hassle he has to go through, even to understand, sometimes to see what has been done, right? So this ELSA tool now is being, based on what I read on FDA's databases, is being allowed to be used for the reviewer to assess what has been done, not necessarily as a decision tool. It is a tool to assess what has been done with excipients, particularly in the area of MDE, maximum daily exposure for excipients, right? It is a tool only to understand, not to decide. Decision has to come with human intervention, right? And again, these tools together with ELSA now basically reflects our field's movement toward a exposure-based and age-specific decision-making. I think the ELSA tool will be very, very helpful, and I think I'm also looking forward to that session to understand. And again, this is the last slide. And again, I think this is a very interesting slide, and that's why I wanted you to hear some of the history, because history shows hidden formulation risk, and then that hidden formulation risk leads to harm. Harm leads to scrutiny, and scrutiny leads to new standard or new regulations. And again, when you look at pediatrics population, particularly, the excipient selection must be justified by age, exposure, the formulation need, and the benefit risk context. Cancer versus cough medication, there could be significant benefits for cancer versus cough. And the goal now is, how are we able to design age-appropriate formulation? This still remains a huge challenge, and that's why you see this group driving that initiative to ensure that maybe in next few years, maybe in next few decades, we will have tools available to formulate, to select excipients based on the formulation needs, or we might need to have some very unique ideas that iCare presented yesterday for some very small patient population as well. Again, we have to remember that we have to use a better judgment. And I was talking to Daniel yesterday and also Preeti, how do we convince and how do we work together as an excipient company, as a drug company, and also as a parent of a child to see that there are medications developed for children? Because believe me, there are 80% of the drugs, even today, they are based just on doctor's intuition, not based on any clinical studies. And that is very scary once you know that, as a father, as a mother. And we need to change that in the future. And I'm sure this initiative was basically initiated exactly with the same goal to address that issue. So I would like to congratulate all the team for organizing this session and also expect questions, during either Q&A or later when we meet. But thank you, and I appreciate your time here.
Make your folder.
Next, we have April Braddy. Going to talk about ELSA. Works at the FDA and in a division that has a lot of abbreviations in the name. So without further ado.
Good morning. I'm April Braddy. I'm a division director at the FDA in the Office of Generic Drugs. I have the privilege of talking to you today about safety of excipients in the pediatric population. My presentation is actually going to sit in the intersection of pediatric drug development and approval, drug excipient safety, as well as assessment tools. So my presentation is kind of broad. It covers several areas. Okay, as a disclaimer, my presentation represents my own views, not the views or policies of the agency. So I want to set that stage. The agenda, I will cover 5 areas. I will start off by talking about excipients from a regulatory perspective and why they matter. I will then go into talking about the assessment process for both innovator as well as generic drug products. Then I will go into talking about our current inactive ingredient database, as well as pediatric considerations in that area. And then I will talk about ELSA and close with key takeaways. So an excipient. Within this, an excipient, as previously shared, the term excipient and inactive ingredient will be used interchangeably. But what I will say here, and this is a thread throughout my presentation, is an excipient is anything but inactive. So when you're thinking about an excipient, it is not inactive. As per the Code of Federal Regulations, an inactive ingredient means any component other than an active ingredient. As I stated, excipients are more than inactive ingredients. Excipients play a critical role in both function and functionality, as well as they can have potential clinical implications as it relates to the overall drug product. So you really have to be mindful of the excipient that you're selecting to be a part of your formulation. Some of the examples of excipients include fillers, diluents, flavors, coloring agents, and so much more that can be a part of a formulation. Some of the critical functions of excipients include delivery, stability, palatability. And as I stated before, because some excipients may have clinical implications, you cannot just assume that they're inert, and therefore rigorous safety evaluation must be involved when you're talking about excipients, right? Oftentimes people say, oh, well, let's just focus on excipients of concern. But that's not the way to think about it, because the level of an excipient can introduce safety concerns, right? So why excipient safety matters? My colleague, Dr. Alexander, spoke about this a little bit earlier, and previous presenter also talked about FDA history as it relates to safety. So I won't go into too much detail about this, but here's just some examples that show that safety concern as it relates to excipients are real. It is not a theoretical concern. These are real concerns when you're talking about excipient safety. You're talking about examples, diethylene glycol, benzoyl alcohol, polyethylene glycol. just to name a few. So as per the Code of Federal Regulations, the definition of a pediatric population, pediatric patients, are broken into 4 groups. So you have neonates, infants, children, adolescents. It starts from birth to 16 years of age. One critical point that I want to emphasize here is through the laws that are currently in place, the FDA fully supports the development and approval and research initiatives as it relates to pediatrics. So, as stated previously, excipients become a very critical component when you're talking about the formulation of a drug product, right? And so when you're thinking about that, there are age considerations as a part of that, and looking at the overall functionality of that excipient, the clinical implications of that excipient, and its age appropriateness. Because in looking at the different age groups for pediatric population, you have to factor in that there are physiological differences. So depending on the excipient, how that excipient is metabolized or tolerated may be impacted based off of that age group. So you can't take for granted that something that's indicated for an adolescent will be safe in a neonate or in an infant. I will now go into talking about how we assess, and this is a very high-level overview, innovator and generic drug products when you're thinking about pediatrics and when you're thinking about excipient in a formulation evaluation. So I want to spend some time here, so bear with me. And as a part of the new drug process, you also have INDs, but I'm going to talk about NDAs as it relates to innovative drug products, and I'll be talking about ANDAs as it relates to generics. So the first consideration is for innovative drug products, the basis of approval is safety and efficacy data. This is based off of both non-clinical and clinical studies. When you're looking at generics, we rely on the safety and efficacy from the innovator, as well as what we define as sameness, which includes same dose, same route of administration, same dosage form, same labeling, as well as establishing bioequivalence. When we talk about bioequivalence, we're considering in vitro or in vivo studies, which can be pharmacokinetic studies, pharmacodynamic studies, or clinical endpoint studies. So these are all factors that are considered when you're talking about innovator versus generic drug products and how they're approved. And when you're talking about pediatrics, right, as previously stated, often for innovative drug products, the ROD is approved and then they come back for that pediatric indication, right? That's not always the case, but that's more than likely the case as to what happens, right? And we have the FDA has certain acts in place, Pediatric Research Equity Act and Best Pharmaceuticals Children Act. So pediatric studies are requested and required as applicable per those regulations. However, for generics, it is unethical to conduct any type of pediatric studies or ask for any type of pediatric studies when you're talking about approval for a generic drug product that may have a pediatric indication. So when you drill a little bit further and then you begin to talk about excipients, there's a broad, sponsors for innovators have a broad choice because they can select the excipients that they want to use in their product. However, factoring in safety and efficacy considerations. For generic products, this is actually limited because when you're thinking about it, you have to make sure that that product performs the same as the innovative product. So therefore, the selection of an excipient for a generic drug product is limited because you have to think about whether it's going to impact the bioequivalence, it's going to impact the safety or efficacy of that product. And for that reason, for certain drug products within the FDA, we actually have regulations in place to where the product has to be Q1 and Q2, meaning qualitatively and quantitatively the same. And those include like optics, ophthalmics, and injectable drug products, right? Factoring in that it is very critical to ensure the safety of the product, right? I know previous presenter talked a little bit about novel excipients, and it was spoken about yesterday. However, novel excipients are still something that is allowable. It may be difficult to get them approved, but they are allowed. Whereas for generics, they're not allowed, right? And when you look at the overall formulation evaluation for innovators, age-appropriate formulations can be developed. For generics, it's one formulation. That's it, just one. And so when we're evaluating that formulation, we're looking at the youngest age because we're taking a risk-based approach as it relates to the evaluation. And as previously stated, the labeling for the generic has to be the same as the ROD. I would spend some time talking about labeling, but that's not the purpose of my presentation. But labeling does become a very critical component. So people may say, well, why does it matter to generics versus innovative products in our assessment process? Because when you think about it, once a generic is approved, insurance companies tend to shift from that innovator to that generic drug product. Therefore, the safety of that generic is just as important as the safety of that innovative product. So we don't stop at the review and approval. Excipient considerations is throughout the life cycle of that drug product because you can have formulation changes, you can have a addition of a new indication, changes in manufacturing. So all of that plays a pivotal role in still considering the excipient that is within a formulation, right? So post-marketing surveillance is throughout. And also there may be new safety signals that are introduced that you may have to consider. So now I'm going to talk about the inactive ingredient database, which currently we do have one available. It provides the maximum daily exposure and maximum potency per unit for approved drug products by the FDA. I'll also get into some limitations as it relates to that. The utilization of the IID and how it's relied upon, and this is just a general context, irrespective of the patient population, for the innovator is one reference point among others. So there's other information that may be used, if that's internal to the agency or that's external literature. It was mentioned that the STEP database may be a resource. This is the same with generics, but it's a little bit more emphasis as it relates to the justification. So it's relied upon a little bit more heavily when you're looking at that data as it relates to a generic drug product. So when you're talking about, again, the excipient review, it is integrated into the non-clinical and clinical program, right? Whereas for a generic, it is conducted based off of the formulation data. Like I stated before, there's one formulation. It's not age appropriate, just one formulation for a generic drug product. So for a generic drug product, an applicant can submit a control correspondence and they can ask the FDA, what I have the XYZ excipients in my formulation, is it acceptable? Or during their pre-drug development, they can ask the FDA, can you review my formulation? Or as a part of their ANDA submission, then they can ask the FDA to review it. So it's a part of multiple steps can be a part of multiple steps, but the fundamental and foundational evaluation from a generic is based off of that formulation information. So here's just a screenshot of the current IID. So this is what it looks like if you were to go to it right now. This is what the IID would look like, and this is how the information will be presented. So with that, there are benefits, there are limitations. So the IID has been around for about 40 years in some form. Previously, it was paper back in 1987, and then it transitioned to an actual form of public database of what you see today. But there are some limitations, and those limitations are specific as it relates to pediatric safety information. And taken directly from the draft guidance, it says, this reflects approved products not necessarily optimal or safe for all populations or all usage and routes. It may not provide different exposure models, for example, safety in pediatric population. So the FDA acknowledges that our current IID does not have safety information as it relates to the pediatric population. Keeping that in mind, then you say, okay, what has or what will the FDA do about that? And we are aware of the feedback that has been given and the ask that has been put to the agency for several years now for an IID that is pediatric specific, right? So the agency is aware of that ask, and it has been shared within public forums, the fact that we have an internal pediatric IID. So this is something that has been shared at SBIA, a recent AAPS meetings of this particular database. This database, this internal resource, I'll refer to that, has supported the safety evaluation, and it has approved it for the agency. It has allowed for us to actually calculate the maximum daily intake and maximum daily exposure for individual ages. As previously stated, age groups are very important, right? So having this individualized data becomes a very useful tool as a part of our assessment process within the agency. And also, we've been able to identify it for multiple dosage forms. So then the question becomes, how could something like this benefit, should it, if it will be made available to the public? And again, the agency understands the ask, and we realize that something like this would actually fill in pediatric safety data gaps, right? And also support research and development and approval. And just in general, providing useful information for patients, caregivers, and physicians. And another critical aspect in recognizing that the potential of a pediatric specific IID would be useful is being able to evaluate the cumulative exposure. Because I listened to the parents on yesterday, the mothers on yesterday, talk about their children taking multiple medications, right? So then you have multiple medications with excipients in them, and you will potentially be able to see if those excipients are throughout the medications that are given, and to assess the risk of that exposure. So now I'm going to walk through two hypothetical scenarios as it relates to the current IID. The first one is an oral tablet, 4 milligrams, chronic population is adults and pediatrics. Youngest age group is 2 years old. So you have your drug X, you have your excipients that are outlined in the table, and then within that table, the MDE value based on the current IID is actually outlined. What is missing from this table is the unit of excipient, and it's purposely missing from this table because you want to ask yourself, and just looking at the IID, knowing what those limitations are, is this acceptable for the pediatric population? That will be a developer question, right? And it will also be an agency question, right? And you really wouldn't know the answer to this until you're developing or as a part of your portfolio, you have a drug product that was previously approved, same route, same dosage form, that may give you some idea, but otherwise you would not know. The second scenario is about weight-based considerations. And this is also a very critical part of our evaluation process within the agency. So when we're looking at the RD labeling, or the RD labeling is being developed, depending on whether they have the age identified for the pediatric population or the weight identified for the pediatric population, that impacts how we're able to calculate the maximum daily dose and the maximum daily exposure of the drug product. So here, again, this is a hypothetical example. The threshold is hypothetical within this example. This is an oral suspension. Maximum dose is 10 mils twice a day. Excipient Z is 5 milligrams per mil. So the population is adults and pediatrics, youngest age group is neonates. So when you do the calculation, based off of that age group, that excipient within that formulation may not be safe. For a child, it's safe. For an infant, it's at the borderline. For neonate, it's not safe. So weight also becomes a critical factor when you're talking about evaluating excipient safety within a formulation. So now, several people have mentioned ELSA. So we'll talk about ELSA. What I will say about ELSA is there's no link to Disney or Frozen or anything like that. It's just a name that the agency came up with for our artificial intelligence tool. So ELSA was launched back in June of last year. And I can actually speak in utilization of this tool. It's pretty great. It's pretty awesome. The current state is pretty good. It is internal to the agency. It's within a secure government cloud environment. So it's used to help us with our current core functions and reviewing work, gathering information, part of our evaluation. What it is not used to make any type of final decisions or draft any type of external communications. Also, what it will not do is replace FDA staff. Our jobs are secure. It's just meant to support us in our review work. So there are two review features of ELSA that is very useful when you're talking about assessment, evaluation of excipients. One is the chat box, which is individualized, where you're able to calculate the MDD and MDE for a particular drug product using ELSA. And then we also have something which is we've been working with over the last couple of months, known as agents. It was previously called Workspace, but this allows for bulk analysis of information as it relates to an excipient, in calculating the maximum daily dose and maximum daily exposure. And so although it takes a little bit more time, it's still a very great tool. And also another thing that's really great about the agent is you can share it with your FDA colleagues. So then you're able to transpose what you've developed and share it with others. So it makes the work more efficient and makes the work more efficient and more consistent as it relates to how we're leveraging AI. So ELSA considerations. There are several factors that are critical to the success of ELSA. We'll talk about two of those. Prompt specificity. You have to be very clear in your prompts. You know, people know this is using AI. You have to be very clear what you're asking for, and you have to verify the results, because ELSA, like other AI agents, will make it up. So you have to be very critical as to the information that you're obtaining from this tool. So here I'm going to walk through an example of a successful ELSA run where we used it to calculate MDD and MDE calculations. So this is the actual prompt. So the information that I'm showing you on screen is actual information that was put into ELSA and that was generated from ELSA. So this is the actual prompt where we asked ELSA to calculate the maximum daily dose of drug X. This was a weight-based calculation. And so as a part of ELSA, we also introduced documents. So again, the RD labeling becomes very critical. Like I said, I wouldn't spend time talking about that, but RD labeling and the information that is in the RD labeling is very critical information as to how it is laid out as it relates to the dosage and administration in order for ELSA to be able to extract that information to be able to perform these calculations. So you definitely have to be mindful of that. Also, as my colleague, Dr. Alexander, mentioned on yesterday, the CDC growth charts. So we also fed that into ELSA as well for ELSA to be able to extract that age-appropriate weight information to be able to do the calculations. And we also introduced the formulation table. So there were 4 documents that we introduced into ELSA in order to be able to evaluate the maximum daily dose and maximum daily exposure. So here's ELSA's output. So this is what it actually looks like, minus the highlights that I put in the slides. This is what it actually looked like coming from ELSA. And then here's the final answer, right? And within that box, it gives the maximum daily dose of drug X and the maximum daily propylene glycol exposure. We validated that information. That information was And then below that, it shows how it was calculated. So this is just an example of how ELSA can be leveraged, as a part of our review process. So I will close with 5 takeaways. Excipients are not truly inactive. Their safety must be rigorously evaluated. The life cycle of a drug product indicated for a pediatric population is dependent upon both the innovator and generic drug products. There are limitations to the current publicly available IID as it relates to the pediatric population. The FDA acknowledges that. A pediatric IID can be a beneficial resource and align with regulatory mandates. ELSA, and lastly, ELSA can be a meaningful tool to support excipient review workflows for innovator and generic drug products when used responsibly with appropriate human verification and oversight. And here's just a listing of resources, FDA guidance documents, ICH guidances as well that help support development of my presentation on this morning. And I would like to thank you for your time and attention.
Sure. A lot of you are thinking of questions as you listen to this. We will have a panel discussion to wrap this all up. Next, we have Ndidi, also from the FDA. And she's going to give her perspective on evaluating excipients. Thank you.
Thank you. Thank you for that introduction, and good morning to all of you. It's been an honor to really be at this workshop. It's been really an eye-opener, and to just be in the midst of so many wonderful researchers that have been doing this work for so many years and decades. It's an honor. As Steve said, my name is Ndidi Nwokorie, and I'm a medical officer in the Division of Pediatrics and Maternal Health in the Center for Drug Evaluation and Research in the Office of New Drugs. So a little different from my colleague, Dr. Braddy, who's in the Office of Generics. I'm in the Office of New Drug Development. So DPMH, as our division is often called, is often the division that is consulted when there is a safety concern regarding an excipient in a new drug. So we're not consulted for generics. That's Dr. Braddy's office. But in new drugs, when there's an excipient safety concern, DPMH is often the division that is consulted to weigh in. So my goal really this morning is to walk you through, how we at DPMH currently approach excipient safety. Again, I have nothing to disclose. This presentation is strictly my perspective and does not reflect the FDA. So we've heard some good definitions of excipient, and I will just briefly go over my definition of excipient to make sure that we're on the same page as I go through my presentation. I will also mention some common excipients of concern, discuss the utility of the inactive ingredient database as a tool in determining excipient safety, and we've heard quite a bit of that, and I think that you're going to see a common thread in the 3 presentations already. And then I will give an overview of the current framework DPMH uses to assess excipient safety when questions arise. And lastly, I will highlight this framework by reviewing a few hypothetical cases. So we know that excipients are inactive substances, as we've talked about, in a drug product added to improve taste or enhance stability. They may help control the release of the drug or act as a lubricant, solvent, or preservative. We know that excipients are not intended to exert therapeutic effects or augment the active pharmaceutical ingredients' pharmacological action. However, as we've heard this morning, despite being inactive, excipients can really pose safety risks, particularly in vulnerable populations such as neonates and young infants. So my presentation will focus on assessing excipient safety in drug products proposed for pediatric use. So several excipients have been identified as posing risks in pediatric populations, and more are being flagged as this field evolves. This list represents some of the commonly flagged problematic excipients we regularly encounter, though by no means is it exhaustive. I have included a reference at the bottom of this slide that have a more extensive list for those that are interested. However, we understand that the effects of a substance are not just qualitative, but also quantitative, meaning our goal should be not only to identify the substance of concern, but the amount of that substance that would be considered safe in a given pediatric population. Ideally, this should be undertaken early in the drug development. Historically, though, requests to evaluate excipient acceptability in pediatric drug products have largely involved adult formulations being repurposed for use in pediatric populations. Or what we often do find is that sponsors developing a 505(b)(2) product who intend to rely on bridging their product to the RLD in lieu of conducting clinical studies may contain excipients in their proposed formulation not present in the RLD, raising additional questions about the adequacy of that bridging strategy to support a finding of safety in pediatric patients. This presents a significant challenge determining whether a given excipient or the cumulative excipient load is safe and appropriate for the largest pediatric population, who, as we know, may have markedly different physiological tolerances than the adult population from whom this formulation was originally designed. So in general, the FDA approaches inactive ingredient assessment by first comparing the inactive ingredient to previously approved levels, searching the literature for safety, or for novel excipients, and in certain circumstances may require additional toxicological data. Sponsors frequently, as you've heard, use the inactive ingredient database to support safety of their inactive ingredient in their drug products. And as we've heard, the IID lists excipient levels present in approved drug products. So if an inactive ingredient is already listed in the IID at or above the proposed level in the novel product for the same route of administration, it is generally recognized as safe in adults. But using the IID to justify excipient use in pediatric formulations, as you've heard, is inadequate, as the IID neither reflects the maximum daily intake of the excipient, nor captures the treatment duration of the listed product, two key factors that are critical in pediatric safety assessment. Furthermore, the IID is agnostic to age of the approved patient population and is broadly understood to reflect the maximum excipient exposure on a per dose basis in adult approved products, making direct application to pediatric populations scientifically unsound. Importantly, the IID does not incorporate any risk-benefit evaluation specific to pediatric patients. So FDA guidance for industry states that in certain cases, sponsors should provide evidence of safety at the proposed level or for the proposed route of administration, taking into consideration the context of use. The Food, Drug, and Cosmetic Act lays out FDA's evidentiary my slides are a little off. this. Okay. So the food drug, this is an old slide deck. The Food, Drug, and Cosmetic Act lays out FDA's evidentiary standard of approval. Pediatric product development is held to the same standard as in adults. Therefore, products approved in children must demonstrate substantial evidence of effectiveness and provide clinical benefit. Adequate safety information must be included in the application to allow for appropriate risk-benefit analysis. With this framework in mind, developers should provide evidence demonstrating that their drug product, including its excipients, is safe for the target population, with excipient safety evaluations included as part of the overall safety submission to allow for adequate risk-benefit assessment. Because the IID does not answer the questions that matter most when evaluating excipient safety in pediatric patients, DPMH takes a different approach by conducting individualized benefit risk analysis for each excipient of concern on a case-by-case basis, take into account what is known and unknown about the excipient in question. So the framework for this excipient safety evaluation, this slide, this evaluation is conducted through a stepwise approach that includes, identifying and clearly defining what the potential safety concern may be. In many cases, available data may be insufficient to inform this risk. However, there may be a strong biological possibility of risk based on the mechanism of action and the metabolism of the excipient. DPMH conducts a comprehensive review of both the data submitted with the application and relevant publication of published non-clinical and clinical literature to characterize the excipient safety profile in the population of concern. We also do look at a worst case scenario. And in order to determine a worst case scenario, we come at this a little differently from the generics, Office of Generics. We calculate a maximum daily dose of the excipient in the full pediatric age range for which the product is intended, using a 95 percentile body weight for each age group. This approach identifies the highest probable excipient exposure a patient within a given age group could receive, thereby ensuring that the safety assessment is protective of the heaviest children at each developmental age. This excipient MDD is based on the MDD of the active pharmaceutical ingredient. Once the predicted MDD is established, the risk of the excipient exposure is weighed against the therapeutic benefit of the drug product in the target population. If the benefit risk is determined to be favorable, additional considerations are assessed, specifically whether early signs of toxicity would be detectable and monitorable, and whether any resulting harm would be amenable to treatment or reversible with drug withdrawal. Following this stepwise assessment, DPH may require the sponsor to conduct additional clinical trials to further evaluate excipient safety in the target population to generate necessary safety data to support product approval. However, when safety concerns are identified late in the product development cycle, approval may be restricted to a subset of the population due to potential risks, with additional enhanced safety monitoring safeguards in place. And in select cases, post-marketing clinical trials may be required to fill outstanding safety data obligations. Therefore, drug developers are encouraged to perform a benefit risk assessment early in the development cycle to identify data gaps and prospectively obtain any additional safety data needed. When conducting a benefit risk evaluation of the excipient, DPMH takes the following factors into consideration: the severity of the disease, or is there an unmet need? Recognizing that a higher degree of excipient-related risk may be acceptable when no adequate therapeutic alternatives exist. Also, is there availability of alternative therapies for the disease in the intended population? as the existence of established treatment options may lower the threshold for acceptable excipient-related risk. Do excipient-free or lower excipient load alternatives exist and suitable for that population of concern? What is the age of the intended population? As younger patients, as we talked about, particularly neonates and young infants, are of greater concern. For example, although a product may contain benzyl alcohol, as we've heard earlier, benzyl alcohol has been associated with some severe toxicity. If approval is being sought for patients 6 years of age and older, the level of concern is considerably reduced, as the metabolic pathways responsible for benzyl alcohol clearance are generally considered to approximate adult capacity by approximately 5 years of age. Other consideration, does the underlying condition have the potential to amplify the excipient toxicity? For example, an excipient that increases GI motility may induce vomiting and diarrhea, and hence fluid loss, thereby exacerbating a disease state in which fluid homeostasis is already critically compromised. So to illustrate how DPMH thinks through these questions using this framework, I will go through a few hypothetical cases. As mentioned earlier, DPMH evaluates excipient safety on a case-by-case basis. In this first hypothetical case, an oral liquid solution is being developed for pediatric use from birth. The sponsor promises this product will provide dosing flexibility over the approved solid oral dosage form that is currently being manipulated to be able to administer the drug to neonates and infants. This oral solution contains a high concentration of glycerol as a dissolving agent. The sponsor justifies the safety of the excipient by citing the IID, which lists products with higher glycerol concentrations than this product, without accounting for daily intake, patient age, or pediatric-specific risks. So the mechanism of concern here is that glycerol is an osmotic agent in the GI tract, where it can cause osmotic diarrhea, leading to fluid and electrolyte imbalance. Systemically, glycerol acts as an osmotic agent and can precipitate intracellular dehydration due to fluid shifts. So when faced with this excipient, the concern is that neonates and infants' GI tracts do not reach adult maturity until approximately 2 years of age. Neonates and infants are particularly vulnerable to these adverse effects, as they're still developing gastrointestinal and renal system, make it difficult for them to regulate fluid and electrolyte, a risk that increases inversely with age. Additionally, conditions dependent on maintaining strict fluid and electrolyte balance may be further exacerbated by episodes of diarrhea. So in this scenario, we will calculate DPH will calculate a maximum daily glycerol dose for each age group at the 95th percentile body weight, assuming the infant receives the maximum proposed API dose per day. This calculation yields the maximum daily excipient load for the heaviest patient in each age group of concern, thereby capturing the worst case exposure scenario, providing for the most conservative assessment for that age group. The outcome of this benefit risk assessment will vary depending on the indication for which the drug product is being developed, right? Early benefit risk assessment in the pre-market phase would identify the safety concern in the youngest, most vulnerable patients, allowing the drug developer to consider reformulating the drug to reduce the glycerol content, to support approval down to birth, or generate additional safety data to provide confidence of the safety of the glycerol content in all pediatric age groups. As stated earlier, when we are faced with these uncertainties during the marketing application phase that is late in development, the benefit risk profile may preclude approval in some populations due to the potential risks. For example, patients requiring strict fluid and electrolyte homeostasis. So the key point here is that early benefit risk assessment in the drug development cycle would allow for greater accessibility to drug products to a broader patient population. Also, disease indication and comorbidities should always be evaluated in conjunction with excipient exposure to determine whether a product is safe across all pediatric age groups or a defined subgroup, a consideration that should be addressed proactively rather than deferred to later stages of development. So also appropriate warning language describing the excipient content and its associated risk should be incorporated into product labeling to ensure prescribers and providers are aware and equipped to make informed treatment decisions and initiate adequate monitoring strategies. FDA's guidance on the incorporation of pediatric information into product labeling provides recommendations on how inactive ingredients of concern should be communicated within the product labeling. Manufacturers of drug products intended for pediatric patients must also evaluate each excipient in their product individually and assess potential excipient interactions early in the pre-market development phase. When faced with questions regarding multiple excipient assessment, DPMH applies the same stepwise approach, evaluating each excipient individually while characterizing the nature and the extent of any interactions between them. To illustrate this approach, consider a hypothetical oral liquid solution being developed for pediatric patients down to birth. This oral solution contains 3 excipients of concern: propylene glycol, polyethylene glycol 400, and benzyl alcohol. So the identified mechanism of concern here is that all 3 excipients are metabolized by the alcohol dehydrogenase pathway, a saturable enzyme pathway. So co-administration creates this competitive metabolic interaction in which the simultaneous presence of multiple ADH substrates can overwhelm the enzyme's capacity, leading to metabolite accumulation that exceeds the risk posed by the individual active single ingredients. Of course, as I've already mentioned, the most vulnerable population of the neonates and young children up to about age 5 who face the greatest vulnerability because the ADH enzyme systems don't actually reach maturity or the adult level until about 5 years of age. So the best benefit risk evaluation here will not support approval down to birth for any indication without pre-market data that addresses these remaining knowledge gaps. Specifically, the potential for systemic accumulation of these 3 excipients in patients with immature metabolic systems. So without adequate pre-market assessment, DPMH may recommend restricting approval to patients greater than 5 years of age where ADH enzymes are closely approximating adult levels, and monitoring, and in addition to monitoring for adverse effects, where just monitoring for adverse effects may serve as a sufficient mitigation strategy. In addition, sponsors should incorporate excipient-specific warning language into product labeling, caution prescribers of this risk of metabolite accumulation from either the combined daily amount of these 3 excipients from the drug products or from other drugs with inactive ingredients utilizing the same metabolic pathways. And as you heard, there are others such as ethanol that also uses this pathway. And as we also heard from our caregiver perspective yesterday, this becomes very important for these children with complex medical need who are on multiple concomitant medications, all of which contain excipients in various concentrations. So early in the drug development, DPMA strongly encourages developers to reformulate the product to eliminate or substantially reduce these excipient combinations, enabling broader approval that extends to younger pediatric patients. Alternatively, developers should generate prospective excipient safety data in the pre-market phase to prevent age range restrictions driven solely by excipient-related safety concerns. So the key point here is when a product contains multiple excipients that share the same metabolic pathway, the combined risk may be greater than the risk of any single excipient alone. Developers and regulators should consider excipient interactions, particularly in young children with immature enzyme systems. So a brief mention of benzyl alcohol. I think our first presenter mentioned benzyl alcohol and pointed out the concerns of using benzyl alcohol as an excipient. And as he pointed out, benzyl alcohol is widely used as an antimicrobial preservative. It's a great preservative, and it's used in injectables and some non-injectable drug products. Of note, it's not used in solid oral dosage forms. So it's really in our liquid formulations and our topicals and our injectables that our babies are getting exposed to. So in 1982, 2 independent reports described 16 neonatal deaths in neonatal intensive care units to repeated intravenous exposures to benzyl alcohol preserved bacteriostatic flush solutions, leading to the clinical presentation termed Gaspin syndrome, which consists of progressive metabolic acidosis, compensatory respiratory Gaspin respirations, and hemodynamic collapse and death. The root cause was thought to be the immature neonatal liver's inability to metabolize benzyl alcohol, allowing for toxic metabolite accumulation. Affected infants were low birth weight and moderately preterm neonates who received doses as high as 99 to 405 milligrams per kilogram per day intravenously. Since then, there have been no comparable cases of benzyl alcohol associated systemic toxicity reported in older infants, children, or adults, or by any other route of administration other than intravenous. So no Gaspin syndrome has been reported in any other population. So the industry and regulatory response at the time were to label intravenous products containing benzyl alcohol, not for use in newborns. However, this population-specific warning has led to a broader avoidance of benzyl alcohol-containing products well beyond the neonatal period, even when exposures were far below the toxic thresholds. So in 2019 and in 2022, DPMH and a multidisciplinary working group conducted a systematic review of the available evidence on benzyl alcohol toxicity. And the group concluded that benzyl alcohol toxicity is clinically monitorable. New onset or worsening metabolic acidosis serves as an early recognizable clinical marker of benzyl alcohol toxicity, making it a detectable and manageable risk that can be appropriately addressed within a clinical development program. The broad population-wide restriction on benzyl alcohol was not supported by the available evidence that we found and was actually preventing pediatric patients from accessing needed therapies. So a revised regulatory approach, we felt, should be to first identify the at-risk subgroup, these preterm neonates, those specifically premature neonates below 44 weeks post-menstrual age, with targeted warnings in subsection 5, warnings and precautions, and subsection 8.4, pediatric use, rather than just this broad population level restriction. And lastly, sponsors should not just exclude pediatric patients from clinical programs just solely on the basis of benzyl alcohol content of their drug product, but instead patients should, beyond the neonatal period, should be evaluated with appropriate safety monitoring in place, with a focus on prospectively generating evidence of safety at the relevant exposure levels to begin to close this data gap, this knowledge gap. So in summary, excipients require pediatric-specific safety evaluation. Excipients are not inherently safe across all patient populations. Pediatric patients, particularly neonates, have distinct physiological vulnerabilities that necessitate dedicated excipient safety assessment independent of adult data. The inactive ingredient database is a starting point, not a safety determination. Inactive ingredient database concentrations reflect precedent, not maximum daily exposure. Evidence, not assumption, should be the standard. Contraindications of excipients in a pediatric product should be based on objective data and not solely on the presence of the excipient in the product. Excipient evaluations should be conducted on a case-by-case benefit-risk framework, taking into consideration the severity of the indication, the patient's age range, route of administration, disease excipient interactions, reversibility of potential adverse effects, and availability of therapeutic alternatives. No single factor should be determinative. Benefit may justify proceeding when alternatives are absent, provided that there's a robust safety monitoring and appropriate labeling are in place to characterize and mitigate residual uncertainties. And then labeling and pharmacovigilance are risk management tools. Well-designed pre-market clinical trials, targeted labeling, and post-market surveillance can address residual excipient safety uncertainty, particularly where adverse effects are clinically monitorable and reversible. And lastly, the regulatory goal is access. FDA's objective is to ensure that pediatric patients can access the therapies they need with risk appropriately characterized and mitigated, and not to exclude products from development based solely on the excipient context. This approach will continue to evolve as scientific understanding of pediatric pharmacology and autogeny advance. Thank you.
We are scheduled for a break and we'll begin again at 10 o'clock.
Right. So yeah, it's me again. And yeah, I can do that, yes. All right, there you go. So yeah, it's me again, but now I'm going to talk about my baby. This is really a topic which is very close to my heart. It is the topic that I did on my PhD on. So yeah, definitely very interesting for me to talk about this. So today with Anjali, we will be going through advancing the pediatric safety through the systematic excipient risk assessment. So in the morning, you've been hearing about the excipient, a very nice background from Sandip, who set the stage into history and telling us really where we are going ahead in terms of the excipient safety and how we are progressing on it. So that was really nice to see. It was nice to hear from April as well on how FDA is introducing the ELSA and they're focusing on the safety aspects of it. From Ndidi, the review framework. And what we want to do is, because this is something really we want to do is, yes, we know the challenges. Yes, we know that excipients, we cannot really assume them to be safe in children because they're safe in adults. But what are we doing about it? And I'm so glad to see that, in today's session, we are discussing that. What are we doing about this? So from us, what are we doing on it? We will be sharing that with you today. And we'll really look forward to a very interactive session, something which we can have discussion with you. So then we understand what challenges you are facing when you're developing the products, and we can then work together to come up with the solutions for it. We are going to start and give something as a start for you in terms of the solution, and then we'll open the floor for a discussion with you to understand if this solution is good enough. But Yep, sorry. No, no, you. Thank you very much. Great. So that's where it is. Thank you. So that's where it is. As I mentioned to you, it's going to be a bit of interactive session. So 15 minutes for the background. And I think so Sandip and other presenters before, they have done something. So we will try to condense that a bit. So then we can get more time in terms of the case study discussion, which will happen for 30 minutes. And then follow on with that, we will have a 10 minutes Q&A session to bring the group together and have that discussion together. And finally, 5 minutes for the wrap up and summary. So this is how we see our session going and flowing today. And then what we will be doing, the first thing is understanding really the selection. So yeah, we know now that the excipients can be a bit tricky, but what are we doing and how do we do that? And how do we go on about it and select the excipients carefully? So we kind of understand the selection landscape and what are the key regulatory concerns that we have. Following on to the systematic risk assessment methodology that we have developed. So Anjali will take you through the whole process and the framework that we've developed. And then with a case study on the mini tablet sprinkles, will really show that, how to get that framework in action and then get some inputs from you. So there where it is, when it comes to the excipient selection, this is something which is really very critical and kind of a roadblock for all of us who are developing and into the pediatric drug formulation is how do we carefully select this excipient? What do we really need to do it? And everybody has their own practices. Everybody is doing it in their own ways. So it is not really standardized. It's very fragmented. Some people may consider one aspect, other may consider the other aspect. I know companies have their internal databases that they often refer to. And what we do usually end up in this is people tend to, or the companies do tend to end up using the excipients that they're familiar with, or they have it in their database already. So they won't really open or to the other excipients, which might be helpful as well. But it's just carefully selecting those and considering those. And then they are really closing the door with that. So that's where we want to kind of open that doors for them. No, you can maybe use the other excipients in your database. It's just a matter of selecting them carefully. And how do you select that? That is what we will show you today. So what's the challenges is in terms of the variability of the outcomes is that the assessors apply different standards. And we get quite a lot of different inconsistent questions. So unless, when I say inconsistent, is the questions that will come from the different regulators. So from the EMA, you would have different questions. From the FDA, you would have different questions. So there is no kind of same platform there or same thing. So one thing that may be accepted in the FDA may not be accepted in EMA and vice versa. So this is where we want to see is, can we harmonize this? Can we standardize this? So then the questions can flow well as well. And then there won't be any inconsistency in the questions that we received. So that's there. And what we saw that as an opportunity because it's to establish a consistent, structured, and transparent, and that is very important, a transparent evaluation assessment process. That would be then, it will be easier for the company to take that process to the regulators, and then probably the same questions would be then raised instead of having all different ways, you doing it in your ways, other doing it in different ways, and then having different questions to it. Can we come up with something consistent way to do this? And that's where we kind of got that into that opportunity, and we developed, the first step to that was to develop best practices. I won't say guidance, but yeah, best practices for selecting the excipients into pediatrics. So this was a workshop, and that's when it really started with it. I mean, of course, it started with the STEP database, but using effectively the STEP database into selection, how do we do that? So we had a workshop in 2018 as a part of the EP5 pre-conference workshop, where we had the industry regulators come together and discuss on how they select the excipients, how do regulators evaluate excipients, and that helped us to bring up this best practices document, and this is published on the website. And the good part about it is if you go and look into this publication, it provides you with the different questions that you should be asking when you select the excipients, and how these questions are different when it is adult and when it is pediatric population, how these questions are different when it is established excipient and when it is a novel excipient. So that's why we have actually come up with all this difference that you should be asking yourself when it comes to selecting the excipients. So and this is kind of a snapshot from that paper, actually. What is different? So what is different when we select the excipients for pediatrics? And I would actually like to start from the core of this figure that I'm showing you, is the first question you need to ask yourself, is this really needed? And it is not rhetorical, really. This is something which the regulators, and it's a genuine regulators and the scientific expectation is, do we really need this excipient? Do we need to think about, we need to think about that. The regulatory agencies, they demand or they ask for a strong rationale on using each and every excipient that you put into your pediatric product, and the principle here is very simple, the minimization, reduce the excipient burden. Any additional excipient that you're adding into it, you're adding an additional risk. Okay, so for example, the colors, if it is only for aesthetic purpose, no. So you need to really explain the purpose, the function, and justify why you want that excipient in the pediatric population. So that is where the first question you need to ask yourself, do we really need the excipient? And then once you have justified the inclusion, then it moves into the safety and toxicity. And again, this is where it becomes different for the pediatrics and adults. We cannot, as I said, we cannot really assume the safety and the toxicity data that we have for adults and we apply that, or assume that they are safe in adults. So assume that is the same for the pediatrics as well. No, we need to collect the data specifically for the safety and toxicity for the pediatrics. If there is any juvenile animal data, that is something that you need to look into. If not, collect some data. But then that's where we were going to discuss in details in the framework. But yeah, the start point could be looking into the acceptability daily intake, which are established by the WHO, FDA, EMA. So yeah, having a look into that. And then the other part is to look into the impact on the ADME. So is the excipient affecting the drug absorption? Or is it in any way affecting the physiological development of the child? And that is something you need to look into is with the pediatric perspective. And then the final step really is to look into the suitability and the acceptability of that excipient. So when I say suitability, yes, does it have a suitable test and it's not affecting the palatability of your overall product? So that is something you have to look into. Then the sensitization. So you're not, you need to look into the sensitization risk so that you're not using, the children are really, the children differ within their own subset as well. The neonate, from the neonate to the adolescents, the physiological development is growing, the maturity of the organ systems is growing. So if you're exposing them to this, it might have a risk to the immunological system. So you need to look into the sensitization risk. You need to look into the stress impurities, and if that has any effect on the targeted age group that you're looking into. So these are the specific three really aspects that you have to differently look into when you are selecting the excipients for pediatrics. So what's the gap really, and what is the regulatory context? I won't go into quite a lot of detail because we had some presentations before, but just to summarize what exists. Yes, we do have some guidelines. We have the guideline from the EMA, from EU on the development of pediatric use, which has a section on the excipient, and it emphasizes that the excipient really needs to be looked into or selected considering all different factors. And Sandip mentioned that, even other presenters mentioned that, you need to really look at it with the age group you're using, the dosage, form you're using, the route you're using, the disease condition, severity of the disease condition. So there is a lot of things, you cannot have a blanket thing that, okay, this excipient is safe, I'll go ahead with it. No, there are these factors that you need to take into consideration, and then it will vary according to that. And that is what the section really emphasizes on. And then is the guideline on the excipients, which again emphasizes on the importance of selecting the excipients carefully for the pediatric population. There is the guideline on the excipients in labeling, which lists the excipients and the recognized effects, and then what should go into the labeling for these excipients. There is FDA guidance on the non-clinical studies for safety evaluation. Then there is the ICH guideline, which again emphasizes on the selection of the excipients, careful selection of the excipients for the pediatric population. So yeah, all this is there, and all of them, they have a same theme going around it, that for each and every excipient, you should be doing a benefit risk assessment. For appropriate selection, what you need to do is you need to do the appropriate benefit risk assessment. But none of these guidelines really give you how to do it. How do we really go on about with the risk benefit assessment? And I was so glad today, and I was telling Ndidi about it. I was so glad. This is the first time really I've seen a slide from the regulator where they've presented the review framework on how they're doing the review. And this is really what we wanted to know. We were missing that bit, is how do really we go on about doing the review process? How do we do the risk assessment? So it was nice to see that coming from you. So that again, for me, I can't say we don't know now. Yes, we know some of it. And we're so glad that it is quite aligned with our thinking as well. So thank you very much, Ndidi, for presenting the review framework. But that is what was lacking. And this is where we thought we need to do something about it. Yes, we've got this step database with all this data, but how do we use this data effectively to do the risk assessment? And how do we do this risk assessment? Because that is something is very important. And this is where I will give my stage to Anjali to take you into it. How do we do this? To answer that how. Over to you. Do you want this?
I have it.
Oh. Yes.
Thank you, Smita, for that nice overview and setting me up to address the how part, right? So that's what I will focus on. So this discussion really started back in 2016 when we had the NSERC conference here and continued the discussion in 2019. And it became very apparent that the gap is there. And with IQ, we want to figure out the how part. We realized that it needs to be global. It just should not be US centric because we file global product, we get questions from the agencies globally, so we have to make it global. That's where the IQ consortium and the EUPFI collaborated. We came together and we said, okay, together we have to figure out the how part because all of us recognize that this is complex. There are several factors to consider. The framework was not established back then by the agencies. There were some guidance, but not enough. So our goals was to provide a systematic method for selection and overall risk assessment of the excipient, focusing on the pediatric formulation. It can be applied to any formulation for the adult, but our focus was on the pediatric. And it should not be cumbersome because that's when applicability in the industry reduces. It has to be enabling the rapid screening and prioritization of the excipients. The main principles were it should create value, right? And it should be used as a tool for integral part of decision making. Should be used to address uncertainty, and it's a systematic, structured, timely. By timely, I mean is it should be part and parcel of as the formulators are coming together to think about the formulation design. So think early in the development, and it's a living document so that as the formulation development progresses, the team should go back to the risk assessment, review it, update it, and inform across within the company, the toxicology group, the clinical group, and also inform the regulator. Use that information for type C interaction to get early read from the regulators. So this is what we were working towards, so that it's a benefit risk-based assessment, but allows that all that transparency and inclusiveness, not just internally, but also with the regulatory agency, and it's a dynamic process. So that's what led us to the Pediatric Excipient Risk Assessment tool. It was a mouthful, so we gave the acronym PARA. And now this framework and tool have been out there since last 2 years. So hopefully the acronym is sticking and people are starting to use it. That's what we will discuss during our interactive session. What's the uptake? What is your feedback and so on? And I would like to acknowledge before I move forward is that you can see down here the IQ pediatric sub-team and also the EUPFI sub-team that worked on this. So it really took a cross-ocean collaboration across the industry, across the academia, and also some regulatory input also to get to the systematic PARA tool. We wanted the framework that we established to be thorough. So as Smita mentioned, we didn't jump into it, just our knowledge that the committee was sharing. We conducted dedicated workshop. We've started with the survey, that's the step one, compiled the survey, published it, and also conducted a dedicated workshop on the best practices to understand where the industry is currently, where the regulatory expectation is current, where do we need to go next. And all that was published so that we can use that as a continued dialogue. And the outcome of that how is this PARA framework. I know it looks very complicated, and today we have limited time, so may not be able to go into the details, but the good news is it's all published and anybody can go and review it and feel free to reach out. But just want to focus on the key parts of it. As you can see, it's quite intense. Quite a few things are mentioned. Essentially, it's a stepwise risk assessment broken down into 4 stages. The first stage is risk identification. For the risk identification, again, there are too many factors. So we created the categories so that it's easier for the users to start putting down the information for each of the category. The first category is patient attribute. Now you heard yesterday, today, Dr. Ndidi's presentation, how important that patient attribute is, because it's all context-based use that we are trying to go. So the age range, the disease type, whether it's a severity, whether it's chronic use or the severe disease, or dosing regimen, dose range, those are the typical ones that we normally take into account. But the co-meds and the precondition, what we noticed was sometimes it's not taken into consideration early on. And that causes challenges later on when we are coming to the time of submission and discussing labeling. So we wanted to put that front and center, that as you're designing your formulation, as you're considering the excipient, take that into account, go back to your clinical team, have that discussion. They may not have full information early on, but whatever is available, take that into consideration. That will help to build a robust formulation and the excipient selection. Product and dosing attributes are critical to make sure that you are selecting the right formulation and the right excipient. So all those were listed. And the excipient attributes, the main ones, right? The level, the amount of excipient, what's the ADI, what's the global regulatory acceptability. Most of the products that we make are for global use. So it's very important, not just the FDA, not just the Europe, take into account globally, Japan. You have to take that into consideration. Many times you have the product, but if you cannot market in Japan, it leads to a lot of additional work. So take that global acceptability into consideration. And the very important one is prior use in pediatric product, because we cannot leverage the adult product, right? It's a prior use in pediatric product often is the key one to justify the level, and also you have to have it context-based. Just having a prior pediatric use is not enough. You have to see what was the disease condition, what was the dosing regimen, what is the severity that was used. So it's a thorough assessment on those aspects of it. So all this is listed. Once you collect all that information, you conduct the risk analysis. As you can see, there are several factors. When we put together this framework, we were like, good, we have the nice detailed framework, but the users might still struggle to put this all together. That's why as a part 2, we developed the tool, Para tool. We were like, we want to make it easy for the users, so they have no excuse not to go about this, right? So we have the framework, we created the tool, which again we'll discuss in the case study, and it's a simple traffic light approach. Did not want to overcomplicate it, so we didn't go through the scoring and all those quantitative aspect of it. We just kept it simple traffic light approach. The goal is high level, look at how the information is evolving and have a discussion cross-functionally with your talks group, with your clinical team, and then come up with the risk management plan. You want to be, the outcome should be no risk or low risk, then you have a free check to go ahead and select the excipient and start your development work. But if you are in the medium risk or a high risk category, that's the value, having that discussion early on, so that together, internally in the team, you can develop a plan, a risk mitigation plan. And as you generate that data based on that gap analysis, that's when the fourth step, the risk review, is critical. Because it's not like one and done deal, right? You get more data, you go back to your risk assessment, fill this tool again, and see, and hopefully you're in all green zone. If you're in all green zone, that's great. If there are some yellows, that's where regulatory interaction come into the play. Like Ndidi was mentioning, right? That could be a labeling consideration. There could be a pharmacovigilance consideration. So those are the dialogues to take place early on rather than towards the end. Yeah, so I'm very pleased to see that now the framework that Ndidi presented and what we have put together, it will nicely fit and it will allow the users to meet all the expectation and the usability of it. So the Para tool, mainly it provides a systematic way to evaluate all the attributes that I was talking about in the framework for the proposed medication in the targeted age range. So there are key questions to ask as the users are starting to fill out the information, right? Is it an appropriate dosage form for the intended patient? You start with the dosage form first. You have to make sure you're picking a right dosage form. And then talk about the excipient. So excipient is an integral part of the dosage form selection. As Sandip was mentioning, it's not a secondary factor, it's a primary factor. It goes hand in hand with the dosage form. So are there any concern with the type and the quantity of the excipient? That's where you have to do your pre-literature search before putting it on your desired list. You weed out some of the very problematic one early on. And then once you have your desired list of excipient, you go through this exercise of the Para tool, which allows practical implementation of the framework, get the heat map, and then go into the next part of it. Are you selecting the excipient? Do you need to go to the alternative formulation or excipients? Briefly touching on what are the key elements of this tool, is essentially taking the framework, and systematically capturing all that information. So all the patient attributes, product and dosing attributes, excipient attributes, filter that into these 4 categories. For part 1 is the biggest one is safety and toxicity, which is not surprising because that's the crux of it. And in the safety and toxicity section, you really have to be very diligent, looking into multiple aspects of it, regulatory aspect, acceptable daily intake, prior use, and also other information. That's where the STEP database also come into handy. Are there any known safety issues or so on? So capture everything, be as thorough as possible. Dosing attributes, that helps you the justification, objectivity, what you are looking for. And this tool is for all routes of administration. It's not just for oral, so that's also needs to be taken into account. If it's injectable, if it's ophthalmic, if it's topical, physicochemical properties. And part 4 we added later on, as we have been developing this over years. It took almost 3 years for the whole journey. So it might seem simple, but it really took very dedicated effort. So we did all the part 1 to part 3, but part 4 is also very important. That's where close partnership with our vendors is important, right? How robust is their supply? What are the other impurities in those excipients? Can those cause any harm? So often we focus only on the main excipient, but what about the impurities in the excipient? That's where the supply network has to be looked into carefully. So that's where I take a pause. So hopefully we have given a preview, nice preview of what it is about. Now we will do a case study session to really get hands-on and interactive. For this, we would like to divide the group into 2, A and B. So those who are in the middle row, if they can just roughly divide and shift to the right and the left side. No need to grab your stuff. It's a short exercise. You can grab a pen or a paper if you need, just for half an hour, indulge us, and then let's have an interactive session. Yeah, so while we are setting it up, if you can just allocate yourself to the half of the group right side. I'm talking to the folks in the middle, if you can shift, and the rest half on the left side. I left it, yeah. So we can start distributing. If you can help Smita to distribute that handout to the folks on that side. And we will be passing on some handouts for the discussion purpose. Yes, please. Thank you. Oh, you're going to do it all? Okay.
Yeah.
I got it. Okay. Was I audible? Was it audible?
Hello.
Chandra, I don't know. If you took
Okay,
some pictures
perfect.
of Smita.
I can.
Sorry?
If you took some pictures of Smita and me, if you can take some pictures. Thank you.
Okay, I'm not sure if I know right now only one of them has a mic on. Both. Okay, yeah, so.
And if there's not enough
Okay,
space,
no
we can
problem.
have
So
one
far,
row.
yeah, only one person
towards
has a
there.
mic on. So we'll see if somebody else ends up putting a mic on, but if it does have like feedback, we'll just go back.
If there is not enough space, then we can have first row still okay for the discussion. You can still have the first row.
Okay.
Oh, you're switching over, right?
Okay, yeah, I'll ask Dr. Hoag because I know he has them.
Want to put the question slide? But
Okay, perfect. Of course.
okay.
But let's see if it on.
We want to show the case study.
No, I don't want to show the camera. Just the questions.
Is the presentation you can go to?
Yeah, it's still behind. Yep.
Okay.
Just the online people won't see it because they'll see just like everybody coming up to the
Yeah.
posters. Yeah.
Maybe they should see the case study, no? Online people.
Yeah.
I don't know whether they will be able to participate in the discussion.
No, we can share this if that's easier.
Yeah.
Okay, so you want to just leave this?
You want to start with the question exercise, right?
The short one, that I will do it informally, you don't have to.
Okay.
Yeah,
So you can leave this one.
This one.
No. Actually, this one.
But let's ask the question just on this blank slide.
Okay, fair enough. Yeah.
Because they'll start reading it as soon as we show the
That's what I'm
slide.
waiting.
Okay, I hope everybody are,
So do you want the online people to just see the screen, or do you want them to see,
I think that if they can just see the screen.
you just want them to see the screen?
Because if they don't see the screen, they won't be able to post any question or anything.
Okay.
I'm the supplier.
What's happening?
Yes, because the people there won't be able to hear us, we'll use mic, but I hope we don't distract each other.
Okay,
Oh, and your job is to make sure you go.
the microphone on.
Yeah, might get feedback.
I need to make sure Mike's again is on it.
For the Zoom people, it might like,
Let's try, otherwise we might have to
Okay.
consolidate.
Can you hear me? But yeah, I'm just trying
Can
not
you
to
not hear
disturb
me?
that group as well, so I'll try to.
Okay. Smita, this side is good.
This
Is there
side-
any interference?
If I speak here, do you hear there?
When Smita is speaking, is it affecting here?
There's too much,
You're good?
can hear both people.
Guys, that's good. Size.
much on the.
Yeah.
You're both, like.
I'll turn off mine.
We'll be other room and then you can. Just curious, what kind of development do you guys see?
We make.
Hello, for the online participants, can you hear us?
So we.
Just wanted to tell that we have actually started with the breakout session. So it will be difficult to involve the online participants into the breakout session because it's more of an internal discussion here. So maybe the online participants can join us in for 20 minutes from now, and then we can come back to wrap up the session.
Yeah, genetic is
Sorry about that. Thank you.
focused,
So,
like patients form it.
Mhm.
So it's basically what we have to do for everyone.
I won't be able to use the mic, so I'll try to just speak
Oh.
loudly, yeah. What we're trying to do here is we have given you some handouts. Anjali presented the framework. It's got the idea of the framework. I wanted to understand the framework is what the get the idea. So then you will be able to connect it to more focus. We'll talk about that. So yeah, I think yeah, come on. I'm gonna ask you another question. How do you do not? We don't make it as practical. Okay, what are the lines with Sandip like you're facing? So, at the moment, how do you have, talking about how are you doing work? And how do you?
I think it comes, you're trying to develop, or. So, hey. I think the first thing they would probably do is, first probably do we have diverse supplier? Do we have all the documentation, all the safety data that is needed for the product? And that is one of the primary criteria. And then we look at the thing, for example, one of the studies that we have recently is, we were asked about color. On our product, and we really colors for product branding. That was about identified. Same size, same for multiple doses. And we have a pass around safety. But the colors are really, I think they're simple comparing them. They are multiple types. They're not competitive, some are not. So I think they have asked some attention. I think people are currently struggling to find out like, oh, maybe we should have used them. We should have taken a different route. So I mean, that is a nice.
Is a full batch way, like it's not full.
Widely available on the shelf for you to see.
Sorry, how to
Yeah.
You should flip it. Just flip it in. Practice it. Yeah. You have to wait. Let this go.
Great, thanks for sharing that experience. Let's just quickly cut over the history. Yeah, and then. Can we have a Q&A session at the end? Okay, now how the experience is working and what happens
It's not going forward.
I think she's going row by row.
Okay. So staying with our themes for the conference, we talked a lot about the mini tablets yesterday, all about the dosage form aspects. Let's just touch upon the excipient part of it, right? So in this case study, we are talking about mini tablets, sprinkles, terminology not finalized yet, but you know what I'm talking about. This particular case study, it's for the chronic dosing. Or the mic, sir?
Yes.
Alcohol. Yeah, but now it's used very widely.
Which one was selected? Going to the tool, right? This is how you capture all the information. So I was explaining the case study, but you can see once you take that information, you can nicely lay it out. In addition to all the dosage form regimen, all those things, you talk also include packaging information, especially when you're talking about suspension solution packaging. The liquids can have the interaction, so you should capture that in the tool. And then the most important one is the body weight, because we are talking about the children in a wide age range, and most often the dosing is done by body weight. So you capture what is the lowest body weight in your most vulnerable population based on the WHO growth chart. And I'll show it in the next slide how that percolates into the calculation. And also you have to take a look into where this product is targeted to be marketed, right? If it's for global use, stability also plays factor sometimes in the interaction of the excipient with the product, and then that will have some harmful effect. So you capture all this information in that para tool where the patient and dosing attributes are captured. Not going. So here I'm just for the example showing that the team came up with a hypothetical formulation. Most of the excipients that you see here were there in the adult formulation. To overcome the bitterness, because the API is mildly bitter, the team was considering two options, mannitol or sucrose. So their approach was just add some sweetener in small quantity in the formulation itself. What I want to highlight here is just because the formulation was okay for the adult doesn't mean from the excipient perspective it's okay in the pediatric. You have to do a full assessment of all the component of the excipients. And that's what this exercise is highlighting. So you put on the discussion all the excipients that you have in the formulation, anything new you are adding, planning to add for the pediatric solubility and palatability purpose, and then look at your formulation, convert it into mg per kg per day. Because when you look at the ADI or the MD, it's milligram per kilogram per day. Then you can do a straightforward comparison that your targeted excipient level, is it within the acceptable limit, above the limit, or yellow, or below the limit. And that's what gives you red, green, yellow traffic light approach. Yeah. So this conversion is important. Then only we can objectively do it very quickly. So that's what is highlighted here. Once you have collected all that information, then comes the putting together or everything into the 4 parts that I was showing in the para-truth. So the very first one is the safety and toxicity. As you can see, several aspects are laid out here: regulatory, acceptable daily intake, pediatric use, prior pediatric use, and the other information. So in this exercise, we're just showing for each of the excipients, when the users or the formulators formed all the information, how does the heat map look like? Today, in the interest of time, we won't be able to go into detail, but I will capture the highlights.
Yeah. Question, you mentioned in your previous slide, great guys, does this table include the solvents used in process materials as well?
It's table does it, but to pick up a good point, you do that exercise for the solvent upstairs. Right now, this table does it, it's very easy to. For example, right now, and also the nitrosamine assessment is part of it. So coming back to this exercise here, what I want to highlight couple of things is you look through the regulatory information across FDA, EU, Japan, and you try to collate all that information. You may not find it. If the excipient has a GRAS status, that helps a lot. If not, then you look for the specifically if there is any guidance out there by any agency on the targeted age range. You have provided some product labeling aspect. There is FDA input is also there. So capture all that. So it's not just about the acceptable daily intake. It's like what is regulatory acceptability for a targeted age range. If that information is available, good. Put it in here. Other important one is to find the prior pediatric use. What is often challenging is the product you can find, but what level of excipient is there in those products is very hard to find. And that's kind of trying to find a needle in the haystack. And that's where I think together we have to discuss how to make that more amenable. Now with the AI and everything, by retaining the confidentiality, how can we still make that information available? There is a roundabout way. You go through the patents, you go through multiple sources of information and indirectly try to work your way into it, but it's very cumbersome exercise. And the goal is to make this straightforward, simple, so that the users can use it effectively. We really have to find a way how that information for the marketed product, it's excipient. Always the API is mentioned, but the excipients are only listed. The amount is not mentioned. Yes, go ahead.
So, yeah, but as far as I'm aware, FDA doesn't have policies about that. So that is a very complex thing to take out, and I think it goes back to what the other factors were saying. Which is, what are the other products that have a similar higher amount? I think that's what I type.
And that's what the challenge is. Yeah. Yeah, and that's what I think you have, but it's handful of excipients like the one that are commonly used, those are the listing items. That's not
Yeah, so
that's the challenge.
So those limits,
definitely that is your first reference point, those product rating guidance, and it's like very
higher, right? So I think that.
and then trying to find the buyer product.
But
But the need is there. How can we have the more information available? So those are the information to capture. Yes. Sorry, I thought you were about to make another point.
How does it address the knowledge gap? Okay, so it'll change, like it changed it to would be higher. if you're already, if you are gonna already do, yeah, trials, let's say, and not do what just this. How could you, get safety?
Yeah, I think that's a really good key thing, though, it's a lot of times like we're development. You don't
And
always
the
exactly
challenge is-
know, or, like, you know, so a lot of the times, like, you know, we look at those levels, there's,
Oh, I didn't
I would say, err on the side of caution.
Is it okay to turn it on?
Yeah.
Okay. Where was that?
Sorry. Depending.
Yes, thank you. So for the safety assessment, the sponsor companies are doing it,
But I,
and that becomes part of the submission, but that doesn't get available, publicly available to other companies. So each company has to do it on their own, and that's what becomes challenge. So how the data sharing, information sharing can be made more publicly available, that's what I think together we have to come and discuss. Otherwise, every company is doing it on their own, and some companies have the time and the bandwidth to do it, some don't, and that's what limits the pediatric product development. Uh-huh.
That could be something that is.
That will immensely help. That will immensely help. Yeah. So it was really good to see that great progress have been made, and already the thoughts have been put together for the pediatric, and it will be good to see to come to fruition. Yeah. So that's how this information was populated, but there are other aspects also to capture, and that's where through STEP database and everything, look at the other toxicity stuff. Thank you, Casey.
like, where's?
You capture those, like some of those are captured here, like with the sucrose, there is a carcinogenic effect. With mannitol, there is a laxative effect. So those information you get from STEP database or from the literature, capture everything about the safety and toxicity of the excipient, right? What I'm trying to say is ADI is not enough. Regulatory expectation across globally, prior use in product, any known information, toxicity information, irritation, palatability, any aspects of it, capture it. Other dosing attributes, sorry, I was going ahead. Palatability part of it, because we are talking about developing a product for children, and this case study is for oral products, so palatability becomes key. So in this case study, as you see, the sodium lauryl sulfate bitter part was highlighted. So you're trying to overcome the bitterness of the API, but you also have to be mindful, are there any excipients in the formulation that can contribute to that challenge? So here SLSs also has a mild bitter taste. So that's what was highlighted when the users were doing the case study. Physicochemical properties, supply chain aspects, all are those captured. So if you apply the PARA framework and PARA tool, have all this information filled in, then the next step is the group within the company comes together. And it's not just CMC. You do need to invite your tox group colleague, your clinical colleague, so you can have a cross-functional discussion, discuss what are the findings, and then what will be the next step in terms of the risk management. So in this case study, luckily, no significant risk was identified for the targeted excipient level. It's very important. The targeted level is mentioned on the top, the mg per kg per day that we calculated for the formulation. So all comparison is being done. What is your targeted usage level? So at the targeted usage level, for the desired age range, given the chronic condition, all that has to be holistically taken into account. To make that statement, no significant risk assessment. It takes a lot of assessment across functionally. What the team identified was both sucrose and mannitol can be used from a excipient safety perspective, but each of those excipients have some considerations. For example, mannitol at a very high level can have a laxative effect. But the team was not concerned because the proposed level in the formulation is very low. So it won't reach that laxative threshold, and that was used as a justification. Sucrose also can be an alternative, but sucrose has a carcinogenic property and could affect blood glucose levels. So that's some of the considerations for the team to take into account. So now they take these two options, go into the formulation development based on the stability, manufacturability, those aspects of it, then they can use it to triage further kind of thing. But at least they have the safety part of it, the selection part of it. The other excipient issue that was highlighted was the SLS, the bitter taste. So there was a round of discussion among the team. What do we do about it? Do we replace the SLS because it is a bitter taste? The team discussed it and said against it because it's integral for the bioavailability. And like I said at the outset, this is a BCS class 2 compound. We do not want to disturb the bioavailability because then it leads to an additional PK studies and so on. So the team's approach was, we'll take a collective approach for API bitterness and the SLS bitterness and overcome it through the sweetener measure. And that approach was what they followed on. Yeah. So now we get into the Q&A part of it, and I can toggle back to the slides as we discuss about it. So I would really like us to now think through this case study and what questions come to your mind. Some of the questions me and Smita have put it just to prompt the discussion. We can capture it, our thoughts on the flip chart, so that when we come together as a group, we can share those. Yeah. Yes, go ahead, Didi.
And there is no like multiple ways we can and there are normalized but you're I'm looking at the type of consider the other approach also. So look out, take that that's sort of midday, but so we do right now heaviest weight patient in that age. You're looking at a newborn going to whether they're like 95 percentile or 6 percentile, the load that they had actually still different than explaining on the tablet for excipients. So knowing that know what that highest what's the highest load that a typical patient do that we can is there a way for this look at the worst case?
Yes, we do that. Yeah, and right now I was just showing one example, but each of your weight, age range, you have to look at the worst case. And out of the calculation, whatever is the most worst case, that's what the whole risk assessment should leverage on. Then, yeah, because if that one is okay, let's say neonate, whatever is the highest mg per kg per day is there for neonate, if that is okay, then the assumption is for the rest of the age group, it should be fine. Yes. Yeah. And then you see which one gives you a higher mg per kg per day, because the excipient load will increase as the mg per kg per day of the API increases. Yeah. Yeah, good point. Good point. And I think that's what usually gets confusing in the practicality part of it. So we really have to sit down and look at each of the age range and tease that out. Yeah. All right. So any question you want to pick up? I think first one, what excipient selection process tool, I think we covered in the outset. Sure. Just going on to this case study, does it showcases the usability of the para framework tool? Any feedback on the framework and the tool itself? Will it help? Anything more to consider? First of all, will it help?
Can I just add one thing?
Do you feel that it is usable?
Yes.
The para framework and the tool?
So you do your assessment one,
Yes.
and then as you go along your product development life cycle, don't go, you go back in this assessment tool to check that your basic assumption.
Yeah, and I think that's the beauty of it. It can be customized, right? It's a framework, like how to go about it, conducting discussion. You can certainly add the processing aspects of it, and we'll see in the next steps. Smita and I have been discussing how to now combine it with the STEP database so that it becomes easier for the users to search the information, populate it in it, and then make it an AI-based tool. Right now, we have published this information that creating that tool, each company has to do it on their own. It's a simple Excel-based format right now, so it can be easily recreated. But we are thinking along that line. So as we develop that, we can expand the scope also further. Yeah, and that's why we are looking for the feedback. Yes, Mona. Yeah, and that's why we were very pleased to see that the thinking is aligned, the framework is aligned, and that will really help the confidence for the users to just go ahead and use it for engagement with the agencies. Yeah. Thank you. Yes, Amy. Okay.
I was curious, is this data always available?
Yes, it's I did it.
Right. But that does not necessarily differentiate adults versus.
I can't remember what information is on there.
Yeah.
Yeah, that's a good point. I'll make a note so that when we are talking about enhancing, so impurities in the excipients. Impact, in this case, on nitrosamine, right? Because that's the key safety aspect that is, we know we all have to first assess and justify. Any other thoughts? I know we are running out of time, so we will have a Q&A session right after it, so we can continue some of the discussion. So maybe one last question I can take? Yes. Yeah.
Well, an assessment, you really need all these excipients. So with the assessment tool, is that decision made for your treatment tool, or do you look at the heat map and start thinking, drop off?
That's a good point, right? If you're leveraging an adult formulation, this heat map, if you find a severe challenge, let's say in the parenteral, polysorbate 80 is very common, right? But depending on what level you're using and how you're going about, if you find a significant risk, we should ask that question. Do we really need that? But can there be an alternative? So it can be used in tandem. But of course, as a starting point, to keep it simple, the formulation, so that this exercise doesn't become long drawn out, we start simple. But if you're leveraging any existing formulation, you should ask that question. Do we really need this excipient? Do we need to spend time on it? Great questions. Sorry, we had limited time today. Normally, we do this workshop in 3 hours. So then we really get into the details of it. Today, we were giving you just a snapshot, but all questions and inputs are welcome. Smita and my information is there. Feel free to reach out. We always love to hear feedback and ways to improve this further. So thank you for your participation and engagement. And we'll continue in the Q&A session. Let's see if Smita can wind up for the wrap up. We have one slide for the wrap up. This is the wrap up. We shouldn't.
Yeah, because it's too long.
Yeah.
And I couldn't hear most of what was said.
Yeah.
Good and learning.
Smita, let's do the wrap up quickly. And then we'll go into the Q&A. I think the splitting up did not work as well, right? Yeah. Because we lost a lot of time. It was okay.
Very good discussion.
Yeah. Discussion was good.
Some important facts that additions that.
Yeah, because it's supposed to end at 11. We're a little, it's not bad.
Yeah, just give us 5 minutes. All right, everyone. We just need your 5 minutes attention for the wrap up.
All right, thank you very much, everyone. Yeah, we're just running over the time, so we will try to wrap this up as much in a small session here. So for the wrap up, I would like to just kind of emphasize again that the PERA framework is published, the tool is published. And yes, someone in our group did ask, where do we get the access to the PERA framework from and the tool? So yes, it is available on the EP5 website.
Yeah.
So you can get the access to the tool. It's a spreadsheet, which you will get the link to from the EP5 website. So please, I would encourage you tomorrow to go into a company and have a look into the tool and see if you can use it. We really would like to welcome the feedback on this tool so that we can improvise it and make it more useful for you.
It's there on the IQ website also, but I think it will have restricted access, right? Only the current IQ users can have access. So UP5 website is your best bet. Otherwise, the publications are listed here. You can access it and it's very easy to create on your own. It's a simple Excel file.
Great. So yeah, maybe just a quick fitting in the information that we discussed in our group. 2 things which we did come up with was one question which was asked is, does the PERA framework consider the chronic use of the excipient. We look into the limits and all, but what if it is used chronically? And that is something specifically we can go and look into it on how we can specifically, yes, the information is there in the tool, but we can make it more specific on how do we capture the chronic use of the excipient into the Ferra tool. And then the other one was what additional elements can I have in the para is the manufacturability. But again, it is a bit challenging because we do have different grades of the excipient. So having the processability and manufacturability information in it might be a bit overwhelming. It would be good to focus on the safety. Yes, we do consider the physicochemical characteristic, interaction, stability, and everything. But having that manufacturability aspect, we will have a look into it and see how we can accommodate into it. But we know that there are some databases. One way could be just linking them to those databases instead of replicating the efforts on it.
Yeah, just to add, there is a supply section in that tool. You can add those aspects of it, anything vendor-specific manufacturability aspect of it. One important thing I want to highlight in our discussion in this group was talking about linking it with the nitrosamine risk assessment. So the impurities in the excipient, can they lead to a nitrosamine risk? I think that was a very good point. So that can be also added on to the para tool. So Smita and we will take that back. And then other one was during the manufacturing of the dosage form, if any solvent is used, let's say for spray drying, organic solvents are commonly used. How do we take that into account? Because they are kind of a process excipients, kind of, and will they impact any safety part of it? So those were the good points that were raised. Thank you for your active participation and engagement, and we'll discuss it further. Smita, do you want to talk about our next steps?
Oh, yeah, sure. So as you all know, we did in our group, we did mention to that this is at the moment a manual spreadsheet that you have to put everything manually and do the greens, reds, yellow, all manually. And we are working towards digitalizing it. So then it will be done automatically. Once you put in the information, it would go green depending upon the information that's available, or it will highlight the risk in red and so on. So that is the next step, really. The next very quick step is to digitalize the para tool. And then the future is really bright. We are working towards the pediatric safety. So we would like to integrate, and that's where the work has already been started and it's in progress. We've completed the phase 1 of it, is where we are leveraging really the AI ML aspects of it to advance and expand the STEP database. So it will have more excipients into it now. So we're using the AI to add the excipients into it. But the ultimate aim really is to have an AI tool which will help you to do all this risk assessment and give you the answers. Yeah, what excipient I can use? What are the safety risks into it? What amounts it has been used? So we are working on that to come up with this AI tool, which will, so you don't have to do everything manually, and it will save so much of time for you. So integrating para, integrating STEP, integrating the FDA, we are in discussion, the 2D database. So integrating all key databases together. So then for you, it would be just going into one AI platform and doing your, asking the questions for the selection and risk assessment of excipient. So that's where we are really, and this is just a snapshot of how the future is going to be bright. So this is where we are going to into AI-enabled expansion of the STEP database. So
yeah, and we just hope that it doesn't take another 3 years to get to the next phase, because now we have the grounding work there, nuts and bolts, and it's very good to see that we have a alignment with the regulatory agencies. So hopefully we can accelerate this process, because that's the crux of the matter. Sharing the excipient level in the marketed product, how can we use to build that database? Yeah.
And last not the least, but I really would like to thank NIH, ABL, with the contract support from the RAPS contract support. We are really able to do this. We couldn't be able to do this alone. We really need all of you and all of your input into it. So then we can, reduce the workload for us and do some smart decisions and informed decisions instead of just doing the regular activities every day. So thank you very much.
We are all in it together. Last slide. I know today the time was short. Like I said, please feel free to reach out with your feedback, input, any suggestions, or how can we make it better, the para tool. And if we have not thought about anything, any factors, would love to hear your feedback. Both Smita and I, contact information is here.
Well, we're going to begin a panel discussion with the same cast of characters. So if all the speakers from the last session